Good day, my name is Dr. Kalona Arumugam from the School of Laboratory Medicine and Medical Science. Today, we'll be looking at the importance of basic human genetic research and its application in clinical settings. The objectives of today's lesson is just to get a brief guide of genetics, understand molecular techniques used in basic genetic research, learning about diseases through genetic variation, and applying what we have learned to a clinical setting. Since the dawn of civilization, humankind has recognized the influence of heredity and applied its principles to the improvement of cultivated crops and domestic animals. A Babylonian tablet more than 6,000 years old shows pedigrees of horses and indicates possible inherited characteristics. Modern genetic analysis stemmed from the work of Gregor Mendel in the middle of the 19th century, where he was inspired to study variation in pea plants. Mendel cultivated and tested more than 29,000 pea plants. Lacking the basic understanding of heredity, Mendel observed various organisms and first utilized genetic analysis to find that traits were inherited from parents and those traits could vary between children. Later, it was found that the unit within each cell responsible for these traits was called genes. Almost every human trait and disease has a genetic component, whether it is inherited or influenced by behavioral factors. Genetic components can also modify the body's response to environmental factors such as toxins. Understanding the underlying concepts of human genetics and the role of genes, behavior, and environment is important for appropriately collecting and applying genetic and genomic information and technologies during clinical care. It is important in improving disease diagnosis and treatment. Cells are the fundamental structural and functional unit of every known living organism. Instructions are needed to direct activities are contained within a DNA sequence. The human genome is packaged into a larger unit known as chromosomes. Human cells contain two sets of chromosomes, one set inherited from each parent. Each cell normally consists of 23 pairs of chromosomes, 22 of which are known as autosomes, and one pair of sex chromosomes. However, the sperm and ova normally contain half as much genetic information. Each chromosome contains many genes, the basic physical and functional unit of heredity. Genes are specific sequences of bases that encode instructions for how to make proteins. Although each cell contains a full complement of DNA, cells use genes selectively. For example, the genes active in the liver differ from the genes active in the brain because each cell performs different functions and therefore requires different proteins. Different genes can also be activated during development or in response to environmental stimuli such as infection or stress. Even though the inheritance of traits was conceptualized by Mendel in the 1800s, genes only became known to us in the 1900s. To understand genes, their structure, function, and role in disease, the concept of the Human Genome Project came to light. The Human Genome Project was designed to unfold the mysteries of the human genome and to see inside it. The Human Genome Project, it started in the 90s and was completed almost 13 years later. The project had two major objectives. That was to create a physical map of the human genome and to sequence the whole genome with all its 3 billion base pairs. Other objectives included and identify disease-causing genes, understand the function behind genes, and to make this information available to researchers across the world. They found that human genome contains 97% of repetitive junk DNA content, meaning that only 2 to 3% of our genome encodes for proteins. But conclusively, the Human Genome Project was one of the most challenging tasks, though an important part of world history, because we had sequenced the whole genome, every single nucleotide of the genome. But how exactly did they manage to achieve such a feat? This brings us to our next topic. Let's look at some important tools that can be used in genetic research. We have previously noted that it is possible to conduct genetic research without directly studying DNA. Instead, some of the greatest geneticists had no special knowledge of DNA at all, but relied instead on analysis of phenotypes, inheritance patterns, and the ratios in carefully designed crosses. Today, classical genetics is often complemented by molecular biology to give molecular genetics, which involves the study of DNA and other macromolecules that have been isolated from an organism. Usually, molecular genetic experiments involve some combination of techniques to isolate and analyze the DNA or RNA transcribed from a particular gene. Before we could even analyze our DNA sequence, we need to extract it. Therefore, DNA purification strategies rely on the chemical properties of DNA that distinguishes it from other molecules in a cell. This is their really long size and their negative charge. To extract purified DNA from a tissue sample, cells are broken up by grinding or lysing in a solution that contains chemicals that protect the DNA while disrupting other components of the cell. These chemicals may include detergents, which dissolve lipids and denature proteins. The supernatant, which contains the DNA and other smaller metabolites, is then mixed with ethanol, which causes DNA to precipitate. A small pellet of DNA can be collected by centrifugation, and after removal of the ethanol, the DNA pellet can be dissolved in water, usually with a small amount of EDTA or pH buffer. Note that this process has purified all the DNA within the tissue sample. The purity of the DNA can be detected using spectrophotometry or gel electrophoresis. Now that we have isolated our DNA, what do we do with it? Once extracted, DNA can be used for molecular analysis such as PCR, sequencing, and cloning. The polymerase chain reaction is a technique for detecting, quantifying, and amplifying nucleic acids. The basic PCR mechanism involves the use of an enzyme called DNA polymerase to synthesize complementary strands of DNA from denatured double-stranded template, effectively doubling the original sample with every cycle of the PCR reaction. This molecular copying operates through cycles of thermal reactions enabled by the assembly of biochemical reagents, which amplifies a few copies of DNA to millions. Amplified DNA can be analyzed qualitatively for its presence or quantitatively by amount or sequentially for its genetic code, and is used in downstream molecular applications. It's important to be familiar with the materials and processes involved in PCR, as many other downstream genetic techniques employ similar strategies. So first off, let's start off with our template. This is a sample of nucleic acids that contains our desired target sequence. Next up, we have primer pairs. Primers are synthetic short DNA DNA strand, which complement the target sequence and binds to the single-stranded DNA. DNTPs are required for DNA polymerase, and DNTPs stands for deoxynuclear triphosphates, and they are nucleotide bases: adenine, thymine, cytosine, and guanine. And this is used to generate the new DNA. DNA polymerase is the enzyme that is thermostable and synthesizes the copy of DNAs. There are three basic steps in PCR. We first have DNA denaturation. This is our first stage. The denaturation stage is where our double-stranded DNA is separated into two single strands by breaking the hydrogen bond between the nucleotide base pairs. Next up is annealing. In this step, our primers anneal or bind at complementary sequences of the denatured DNA strand. If the annealing temperature is too high, primers do not anneal efficiently. If it's too low, primers may bind non-specifically to the template. The final stage is extension and occurs at a temperature that allows for the optimal polymerase activity of binding nucleotides to the annealed primer, resulting in an exponential amplification of the template strand. The process or these three steps are usually repeated for 40 cycles until the initial few copies are amplified to millions of copies. The resulting amplicon can be visualized through staining with a dye or through labeling with fluorescent nucleotides or primers. In an electrophoresis reaction, agarose gel and visualizing molecules demonstrate the size and quantity of the DNA product in comparison to DNA ladders, which is a molecular weight marker with standard lengths and markers. This visualization is conducted in reactions like gel electrophoresis, a standard procedure that separates DNA molecules via their length and demonstrates their homogeneity. Electrical fields causes the DNA to migrate through the gel matrix at different speeds as they are tagged with other molecules for easy identification through color fluorescence. Shorter fragments will move at a more rapid pace, so approximate size can be calculated by the comparison between PCR fragments, and the DNA ladder is used to confirm consistency. Various specialized PCR techniques have been developed to address researchers' specific needs. The use of a specific type of PCR depends on the nucleic acid source, the desired amplicon, and the downstream applications. Here we have 4 different types that are PCR that are commonly used, each with its own advantage and disadvantage. One application in which amplified DNA can be used is through sequencing approaches. We previously spoke about the Human Genome Project and how we determine the order of every single nucleotide base present in the entire human genome. This year marked the 21st anniversary of the Human Genome Project that took a lot of time, innovation, and money. We're talking about $300 million. But now the technology has advanced so far that human genomes are routinely sequenced for under $1,000 in a matter of days. So what is this DNA sequencing technology? I'm glad you asked. Firstly, let's discuss the general steps involved in sequencing before we speak about the 3 different platforms. These steps will of course vary from platform to platform. First off, we start with our sample prep. Our DNA needs to be isolated. The quality and yield of DNA is a major concern in sequencing as it reduces the chances of reaction failure. Next up, we have our amplification step. Amplification is a pivotal step in sequencing. Here, only the gene of interest or the DNA sequence we wish to study is first amplified. After PCR, we need to get rid of the unbound primers, primer dimers, unused Taq polymerase, unused templates, and other unused PCR reaction buffer components, as it can abort DNA sequencing. And this is why purification is important. Sample pre-preparation is a very crucial step during DNA sequencing. During this step, adapted DNA sequences are ligated on both DNA ends. To the adapter, a primer anneals for the amplification. The amplification process in sequencing is a lot like PCR. However, here the nucleotides are either radio or fluorescent labeled. With it, high-fidelity DNA polymerase and other key ingredients are added to the reaction. The prepared reaction tubes are placed in a sequencer machine. During the reaction in the sequencer, denaturation, annealing, and extension occurs simultaneously. The signals of the addition of each complementary nucleotide are recorded by the machine, and the data is sent to a computer. Once the whole DNA is sequenced, the results are saved in a unique file format with inbuilt software to process the data and compare it to an available library or dataset. There are different types of DNA sequencing platforms. First, we'll look at first generation sequencing known as Sanger sequencing. The traditional DNA sequencing method is Sanger sequencing or chain termination method. It may be old gen, but it's really accurate and it's still the go-to if you have a specific region of DNA you want to read. The Sanger sequencing involves making many copies of a target DNA region. Its ingredients are similar to PCR. However, Sanger's sequencing reaction also contains a unique ingredient. This is dideoxy or chain-terminating versions of all four nucleotides. Sanger sequencing involves making many copies of a target DNA region, each labeled with a different dye. Once a didioxy nucleotide has been added to the chain, there is no hydroxyl available for further nucleotides to be added. The chain ends with the didioxynucleotide, which is marked with a particular color of dye depending on the base that it carries. This process is repeated in a number of cycles. By the time the cycling is complete, it's virtually guaranteed that a didioxynucleotide will be incorporated at every single position of the targeted DNA in at least one reaction. That is, the tube will contain fragments of different lengths, ending at each of the nucleotide positions in the original DNA. After the reaction is done, the fragments are run through a process called capillary gel electrophoresis. Short fragments move quickly through the pores of gel, while long fragments move more slowly. As each fragment crosses the finishing line at the end of the tube, it is illuminated by a laser, allowing the attached dye to be detected. The data recorded by the detector consists of a series of peaks in fluorescence intensity, as shown in the chromatogram. The DNA sequence is read from the peaks in the chromatogram. Sanger sequencing gives high-quality sequence for relatively long stretches of DNA, up to 900 base pairs. However, Sanger sequencing is expensive and inefficient for large-scale projects, such as sequencing the entire genome. For tasks such as these, new large-scale sequencing techniques are faster and less expensive. The principle behind next-generation sequencing is similar to that of Sanger sequencing, which relies on capillary electrophoresis. The Sanger method requires separate steps for sequencing, separation, and detection, which made it difficult to automate the sample prep, and it was limited in its throughput, scalability, and resolution. Next-generation sequencing method uses array-based sequencing, which combines techniques developed in Sanger sequencing to process millions of reactions in parallel, resulting in high speed and throughput at reduced costs. The genome sequencing project that took many years with Sanger sequencing can now be completed in hours with next-generation sequencing, although with shorter read lengths, about 50 to 500 bases, and less accuracy. Next-generation method of DNA sequencing has three general steps. We have our library prep. Libraries are created using random fragments of DNA either by sonication or enzyme digestion, followed by ligation with custom linkers that enable the sequence to become bound to a complementary counterpart. Okay. Next, we have amplification, where the library is amplified using clonal amplification methods and PCR. Next is sequencing. The sequencing method is based on sequencing by synthesis, as the polymerase adds the nucleotide into the amplification. The amplification signals are recorded each time. This will generate multiple sequencing databases for the DNA sequence. The read generated by the sequence can be aligned to the reference genome sequence, and by doing this can identify an addition, deletion, or variation in the sequence. So now we're looking at third generation sequencing. Well, Sanger sequencing is good at sequencing long reads. It's not high throughput. Next generation sequencing is high throughput, but cannot sequence long reads. What if we want to sequence long reads with high throughput platform? Boom, here comes third generation sequencing. The first type is the Pacific Bioscience single molecule real-time sequencing from Pacific Bio. This technique has miniaturized wells known as zero-mode waveguides, in which a single polymerase incorporates labeled nucleotides and light emission is measured in real time. The use of flow cell allows thousands of DNA to be sequenced at the same time. However, the polymerase works super fast. It's hard to see what is added and which creates errors. The errors can be reduced by reading the sequence several times. Next is MinION or MinION by Oxford. Unlike the fridge-sized Pacific Bio machine, the MinION is only the size of an iPod. The small size makes it very portable. Scientists can take it with them from place to place and to places they may not have sequencing sensors. The principle of MinION is different from Pacific Bio. It does not use dyed nucleotides. Instead, MinION is based on the fact that each nucleotide is a different size and has a different electrical property. Nanopore also uses a flow cell, which allows massive parallel sequencing. Here we have some basically just the differences between each of the sequencing platforms. We can see that first generation separates by size, and second generation where we have parallel sequencing of multiple small fragments, and third generation where we have single molecule real-time sequencing. Each has their own different accuracy, throughput, and of course costing. So another technique used by genetic researchers is genotyping. A single nucleotide polymorphism, or a SNP, is a variation at a single position in a DNA sequence amongst individuals. If more than 1% of the population does not carry the same nucleotide at a specific location in the DNA sequence, this variation is then classified as a SNP. If a SNP occurs within a gene, then the gene is described as having more than one allele. In some cases, SNPs can lead to variations in the amino acid sequence. However, not all SNPs are not just associated with genes. They can also occur in non-coding regions of DNA. Although particular SNPs may not cause a disease or disorder, some SNPs are associated with certain diseases. These associations allow scientists to look for SNPs in order to evaluate an individual's genetic predisposition to develop a disease. In addition, if certain SNPs are known to be associated with a trait, then scientists can examine stretches of DNA near these SNPs in order to attempt to identify the gene or genes responsible for the trait. SNPs can be detected using a myriad of techniques. Currently, over 20 different SNP genotyping methods consisting of various combinations of different allele discrimination chemistries and single detection methods have been described. So we have more traditional approaches such as RFLPs, allele-specific PCR, denatured gradient gel electrophoresis, and more high throughput methods such as sequencing using DNA arrays and high-resolution melting. Now, genetics, the study design is extremely important in genetic studies. Generally, there are two approaches. We have the candidate gene association studies. Here we look at specific genes as selected based on prior knowledge from mutational, biochemical, or linkage analysis of a specific trait of interest. It is a low-cost, hypothesis-driven, trait-specific approach, but what the disadvantage is they might miss unknown loci. As attractive as hypothesis-driven candidate gene approach is, it focuses exclusively on relatively few known genes, ignoring many that have not yet been characterized. Our major drawback of candidate gene approach is that a prior knowledge of the pathogenesis of the disease is required. When the molecular mechanism is poorly understood, it could lead to a selection of wrong genes. Now, the next approach is a genome-wide association study. This is a comprehensive approach to systematically search the genome for casual genetic variation. A large number of markers are tested for association with various complex traits, and no prior information on the gene is required. GWAS has the advantage that no prior knowledge of the structure, function, susceptibility of the gene. Hence, this approach provides the possibility of identifying genes that modulate susceptibility to infectious diseases that had previously not been suspected. Okay. So GWAS is used to general workflow of how we would do a GWAS is that a GWAS is used to identify SNPs correlating with the disease risk. Genotyping arrays such as your immunochip can be used for both the replication of your GWAS loci and genetic fine mapping of all variants in disease-associated loci, further narrowing down the association signal. Bioinformatics can then be used to both locate and functionally annotate SNPs in linkage disequilibrium with the lead SNPs. In non-coding regions, SNPs coinciding with regulatory features such as histone modifications or transcription factor binding sites are most likely to have an effect on gene expression. Appropriate experimental techniques can be used to investigate genotype-specific interactions in disease-relevant cell types. SNPs associated with disease that coincide with coding regions of genes require bespoke experimental confirmation dependent on both position within the gene, function of the protein. Once a causal variant affecting gene function has been identified, its effect on relevant biochemical pathways and resultant phenotype can be investigated. Identification of susceptible variants that underlie many disease processes has increased confidence that this information can be translated into clinically beneficial improvements in patient care. There are two principal routes through which translation might be effective. In the first, we identify novel causal pathways, which provides new opportunities for clinical advances of generic benefit to all those suffering from or at risk for the disease concerned. Identification of therapeutic targets can lead to novel therapeutic agents. Identification of biomarkers improves disease prediction and monitoring of disease progression and treatment response. Identification of environmental contributors to the disease enables disease prevention measures. The second translational route lies through using knowledge of individual pattern of disease predisposition to develop more personalized approaches to disease management, including using personalized diagnostics or prognostics to enhance therapeutic optimization. However, variants so far identified explain only a small portion of individual variation in disease risk. Consequently, for most individuals, genetic profiling using currently available markers provides only limited information about disease risk. So we can use genetic biomarkers to diagnose disease, predict the severity of disease, as well as to predict treatment responses. Genetic research is important for developing and identifying new and novel preventative and therapeutic strategies to treat specific diseases. Gene therapy is using nucleic acids to treat genetic disorders. These nucleic acids can be designed in a variety of ways to achieve the same therapeutic outcome. Gene therapy tools can be used to correct a mutant gene by one of three ways: expressing a healthy copy of a gene, silencing or inactivating the mutant gene transcript, and using genome editing tools to repair or turn off mutated gene. An example is gene augmentation therapy. This is used to treat diseases caused by a mutation that stops the gene from producing a functioning product such as a protein. This therapy adds DNA containing a functional version of the lost gene back into the cell. The new gene produces a functioning product at sufficient levels to replace the protein that was originally missing. This is only successful if the effects of the disease are reversible and have not resulted in long-lasting damage to the body. For example, this is used to treat loss of function disorders such as cystic fibrosis by introducing a functional copy of a gene to correct the disease. And the gene they correct is your CTFR gene. P53, which is often dysfunctional in cancer, can also be corrected using this method. Aside from using gene therapy, we have also developed techniques known as gene editing. Gene editing is a group of technologies that give scientists the ability to change an organism's DNA. This technology allows genetic material to be added, removed, or altered at particular locations in the genome. Several approaches to genome editing have been developed. A recent one is known as the CRISPR-Cas9, which is a short for Clustered Regulatory Interspaced Short Palindromic Repeats and CRISPR-associated protein 9. The CRISPR-Cas9 system has generated a lot of excitement in the scientific community because it's faster, cheaper, more accurate, and more efficient than other existing genome editing methods. It has already been shown to be effective in treating muscular dystrophy, Huntington's disease, and sickle cell anemia. CRISPR-Cas9 was adapted from a naturally occurring genome editing system found in bacteria. Cas9 protein complexes with a guide RNA. The guide RNA contains a short sequence that binds to a specific target sequence of DNA in the genome. The modified RNA is used to recognize the DNA sequence, while the Cas9 enzyme cuts the DNA at targeted locations. Once the DNA is cut, researchers use the cell's own DNA repair machinery to add or delete pieces of genetic material, or to make changes to the DNA by replacing the existing segment with a customized DNA sequence. Although Cas9 is the enzyme that is used most often, other enzymes such as CPF1 can be used. Once the DNA is CPF can be used. Okay, so an example in which CRISPR-Cas9 can be used is within HIV. So we know that HIV uses CCR5 to gain entry into T cells. We could edit our CCR5 genes so that they become defective and there's improper binding of the HIV. We could edit CCR5 gene so that it does not express on our T cells, again preventing the binding of HIV. We could also directly remove HIV from our genome once HIV is integrated by using CRISPR-Cas9. CRISPR-Cas9 can be also used to target viral cDNA. Now, there are many challenges with gene therapy and gene editing techniques. Because our cells divide rapidly, the therapy may not have long-lasting effects. Because a foreign complex is being introduced into our body during gene therapy, we might develop an immune response against it. Furthermore, disorders caused by multiple genes cannot be effectively cured by gene therapy. Lastly, genome editing techniques such as CRISPR have many ethical concerns, and as they may be used to has many ethical concerns that need to be taken into consideration. The cycle on the left reflects basic genomic research, while the cycle on the right reflects a genomic learning healthcare system. Note that new knowledge emerging from either the left or right cycle has the potential to feed into the other, creating opportunities for bench to bedside and bedside back to bench progression, both of which are expected to grow in the coming decade. It is exciting to hypothesize how the expanding role of genomics in medicine will impact our understanding and classification of diseases. Ultimately, this will better classify our understanding of illnesses and improve treatment strategies and research. Thank you.